So one of the roles of galactose in the body is actually to be part of a glycoprotein that is basically a marker on our red blood cells. So I'm sure most of you have heard of the ABO blood groups. So what the ABO blood groups are is basically a way of specifying what types of compounds or antigens you have on the surface of your red blood cells. So for example, if you're blood type A, then you have A-type antigens on your red blood cells. If you're blood type B, you have B-type antigens on your red blood cells. You can be AB, so have both types of antigens, or if you're O, then there's actually no antigens on those red blood cells that are recognized via this system. So here we can see a representation of the red blood cell and the glycoprotein, which you can see is this quite irregular shaped component, and the D-galactose would be part of that glycoprotein as well as many other units. But essentially, let's say that this glycoprotein was type A. So that would mean that the individual with this type of red blood cell with the glycoprotein on the surface would be blood type A. So remember, a glycoprotein is a combination of a sugar, which is the glyco part, and the protein means protein. So we've got a fusion between these two different types of component. So basically the blood typing dictates whether you'll be able to receive blood from a particular individual. You also have to take into account the rhesus factor. So some people are rhesus positive, some are rhesus negative. So a rhesus positive individual can't donate blood to a rhesus negative because the white blood cells of the rhesus negative person will start attacking the red blood cells of the other blood type where it has the positive. So a similar thing happens if you have the wrong type of antigen in terms of this ABO blood system. So for example, if an individual had the glycoprotein A type on their red blood cells, like I've shown you over here, and they were given B-type blood with B-type antigens, that would actually mean that the individual with A-type blood would actually have an immune system that would attack this B-type blood, because the individual's immune system is actually recognizing that the red blood cells are not part of the self. So the immune system is engineered to actually attack what it recognizes as non-self. So because the blood doesn't have the same antigens as the blood it's used to, it attacks it and destroys it. So what about if an individual with O-type blood with no antigens was given either A or B-type blood? Would either of the A or B-type blood be able to be accepted by the individual with O-type blood? No, because the individual with O-type blood doesn't have A or B antigens, so that actually wouldn't work. The white blood cells of the O-type individual would actually destroy the new blood. What about if O-type blood was given to an individual with B antigens or an individual with A antigens? Do you think that would work? Yes, it would, because the O-type blood actually has none of these antigens. So there's no way for the individual's immune systems with the A or B-type blood to actually recognize that it's foreign and attack it. So that's why we call O-type blood universal donors, because they can actually donate blood to anyone. The best donors are actually O-type blood that are rhesus negative, because they don't have the antigen of the rhesus factor either, so they can donate their blood to absolutely anyone. It's quite a rare blood type. So the next sugar we're going to have a look at is fructose. So remember, fructose is classed as a ketohexose in the straight chain form because we've got that ketone group in the middle of the structure. And we can see the structure when it's in its ring form here, which is a 5-membered ring. So in both cases, whether it's a ring structure or in this straight chain form, we've got 6 carbons. So fructose is actually the sweetest tasting sugar, and it's why the sucrose, which is our table sugar, tastes sweet. So we have this disaccharide sucrose present in anything that really tastes sweet, high concentrations in fruit and also honey. So sucrose itself is a disaccharide that is composed of one fructose and one glucose, as we can see here. So the 5-membered ring on the right hand side is the fructose, and the glucose, which is the 6-membered ring, is on the left hand side. So how are the individual monosaccharide units connected within sucrose? Well, you can see we've got that oxygen in the middle that's connected to either side. So that's our ether functional group that is facilitating that connection. And we can also call that the glycosidic bond. So the body handles fructose quite differently to the way it handles glucose. The body is adapted to use glucose as a fuel source in both storage mechanism in terms of storing it as glycogen, or to use it in lots of different ways to actually generate energy. However, it's not quite as good at creating energy from fructose. So there is a reason for this, because we're adapted to use glucose, because that was the more abundant carbohydrate, say when we were foragers. You didn't often come across a nest of bees and get the honey that way. You'd be more commonly finding plant material that would have, for example, the fruit growing off it, and that would give you some of the glucose. But fructose, in terms of getting it from our sucrose component, was more rare. So that's why we're adapted to using glucose rather than fructose. So what does the body actually do with fructose once you've digested it? So you've eaten a chocolate or something else that is quite sweet. Well, in the digestion, sucrose will actually be broken down into the glucose and fructose units, and then from there it'll be absorbed through the small intestine and enter the blood. The fructose will then be taken to the liver, where it can be metabolized. So there's actually no storage mechanism for fructose in the body. So basically this means that whatever fructose comes into the liver, it has to be used. There's no way of storing it. Whereas if it's glucose, it can actually be stored as glycogen, or it can stay in the blood. So the way the body uses glucose is much more varied. So what about fructose? If there's lots of fructose coming into the liver, well, there is a mechanism for fructose to be turned into glucose, but once the glucose stores are full and the body doesn't want to make any more glucose, then one of the things that can happen with fructose is it can actually be converted into triacylglycerol, which is essentially fat. So the triacylglycerol is actually going to be predominantly produced in the liver through this lipogenesis process, another one of the pathways that we'll look at. So this is why there's this idea that if you eat too much sugar, your liver turns to fat. Roughly 1 or 2% of the fructose that you ingest when you eat a sugary substance will actually be turned into fat. So if you're eating lots and lots of sugar every day, that is equivalent to putting grams of fat into your liver. So for example, you eat 100 grams of sugar in a day. That's equivalent to putting an extra 2 grams of fat into your liver. And if you're doing that every day for a year, for example, your liver is going to actually become quite fatty. As well as that, some of that fat will actually go out into your bloodstream and increase the amount of fat in your blood. So this is one of the reasons why it's actually quite unhealthy to be eating too much sugar, because some of that is actually going to be converted into fat and it'll be accumulating in the liver and actually causing damage to the liver. There's something called fatty liver disease that's predominantly caused through this mechanism. So another role of our monosaccharides is to be one of the units within DNA and also RNA. So here we can see an individual unit of DNA, and an individual unit of DNA is called a nucleotide. So for example, a DNA helix will actually be composed of thousands of these nucleotides. So we're just considering one individual unit. So within an individual unit, we've got these three components. On the right hand side, we've got the nitrogen containing base, which we saw in the organic chemistry lecture. So this one looks like guanine. Then attached to that, we've got a sugar unit. So you can see a 5-membered ring that has these OH groups and also the ether functional group. So this is a sugar, and it's actually, in this case, it's a ribose sugar. So it can be a deoxyribose sugar as well. So I'll show you the difference between that on the next slide. And we've got a third component, which is a phosphate on the left-hand side here, which is actually one of our polyatomic ions. So phosphates are very common in the compounds that we use for metabolism. So we'll be seeing this more and more. ATP, which is our cellular energy, actually contains 3 of these phosphates. So within these nucleotides, we can actually work out whether it's going to be part of DNA or RNA based on what type of sugar is present within that structure. So you can see on the left-hand side, we've got the ribose sugar. So the ribose sugar has both groups that are down the bottom here being OH groups. So if we compare this to the sugar present in DNA, which is deoxyribose, looking at those same 2 groups there, one is an OH, just like in the ribose, but the other is now a hydrogen, which is different to what we saw in the ribose. So you can see that that's where the difference is in the structures. If it's a ribose, it has the extra oxygen in the structure. If it's a deoxyribose, it has one less oxygen. And that's where the name deoxy comes from. So deoxy basically means one less oxygen. And that's also where the D in DNA comes from. So D corresponds to deoxy, whereas the R in RNA actually corresponds to ribose. So that's a handy way of remembering it. So what type of sugar does DNA contain? Would you expect it to have the ribose or the deoxyribose? Well, it's going to have the deoxyribose because that's the one that starts with D, and hence it's going to be present in DNA. What about for RNA? What type of sugar would it have? Well, it's going to have the ribose sugar with the extra oxygen there, and that's both starting with R, so we've got ribose corresponding to the R in RNA there as well. Alright, so I've already shown you that monosaccharides can exist in 2 forms, either a ring form or a straight chain form. But now we're going to investigate some of the differences between these 2 forms. So if the monosaccharide has 5 or more carbons, then it's capable of forming one of these ring structures where we have each of the atoms in the ring like this. Whereas a straight chain form will have defined ends with no ends within the ring because it's all connected. So here we can see the variations of glucose. So glucose can either exist in a cyclic form, which it's normally seen in. So you can call that the glucose ring structure. Whereas on the right hand side, we've got the straight chain structure of glucose. So there are some differences between the cyclic and straight chain form, rather more than just the fact that they're in these 2 forms. So because it's in a ring structure, it actually changes the structure a bit. So one of the main differences between a straight chain and a ring structure is the presence and absence of the aldehyde and ketone functional groups. So remember earlier on I showed you that when sugars were in the straight chain form, they could contain the aldehyde group at the end of the compound or the ketone group in the middle of the compound if it was a ketose. However, these functional groups are actually absent in cyclic sugars. So when it's in a ring structure, we don't see the aldehyde or the ketone groups at all. So if you want to prove that to yourself, look at fructose and glucose in these ring structures. there's nowhere within these structures that you can see the carbonyl group, and hence neither have a aldehyde or ketone group. So there are those aldehyde and ketone groups when we have the straight chain form for each of the fructose and glucose. But once the sugars actually form the ring structures, they no longer have those aldehyde and ketone groups. Instead, they've got the ether functional group, which is where we've got an oxygen attached to two carbon atoms on either side. So you can see in fructose, the ether is at the top of the ring structure as a part of a 5-membered ring, and in glucose, it's part of a 6-membered ring structure. But in both cases, they have the ether functional group in comparison to either the ketone or aldehyde. So this is the main difference between cyclic and straight chain structures: the absence of the aldehyde and ketone in that ring structure and the presence of the ether functional group. So you can see, hopefully you can see what the ether functional group is actually allowing within the ring structure here. Essentially, it allows the whole chain to close. So you can think of the straight chain structure as a straight line, and the ring structure is kind of like a circle that's closed. So what the formation of the ether functional group does is it allows the ring to actually close. So each side of the ring is actually bonded to the ether, whereas in the straight chain form, there's no ether, so there's actually two defined ends at each of the ends of the straight chain structure. So you can think of the ether functional group as an attachment that allows the ring to actually form. There's a little bit more detail there than you really need to know, but thought you might be interested in that extra bit of information. So we've been through quite a lot of content in the first part of this session, so let's summarize what we've learnt. So we saw for the carbohydrates that they come in 3 forms: single sugar forms, which are our monosaccharides; disaccharides, where we've got 2 sugar units attached together via a glycosidic bond; and then polysaccharides, which are our larger sugars containing between 100 and up to 1 million connected sugar units. The straight chain monosaccharides could contain either the aldehyde functional group, that would make it an aldose, or the ketone functional group, making it a ketose. And we also saw that the straight chain monosaccharides could exist as either an L or D isomer, which corresponded to L being the left-handed version and D being the right-handed version. Spent a fair bit of time talking about glucose, which is the most abundant monosaccharide in nature, and we're very reliant on it as humans. So it's a 6-carbon aldose sugar that the body relies on to generate cellular energy. And we control the amount of glucose in our blood within a fine range using insulin and glucagon. Insulin being released from the pancreas to decrease the blood glucose level, facilitate movement of the glucose into the cells, and glucagon will be released from the pancreas to try and increase the blood glucose by facilitating the movement of glucose out of the cells into the blood. So we looked at some of the characteristics of galactose, which was very similar to glucose in terms of it being a 6-carbon sugar that was also an aldose. And we also saw that it contributed to this ABO blood group system because part of the antigen on the red blood cell surface contained the galactose unit. Fructose was an interesting one because it's the sweetest tasting sugar, and it's actually a ketose containing the ketone group. Even our DNA and RNA relies on sugar units, particularly the ribose in RNA and deoxyribose in DNA. And the difference between these structures being the deoxyribose has 1 less oxygen than the ribose. And I also showed you that if a sugar contains 5 or more carbons, then it can exist in these straight chain and cyclic ring forms. So you can have either form of the sugar. But there were some differences between these because in the cyclic form, the ether functional group was present, whereas in the straight chain form, it could either be an aldose with an aldehyde or a ketose with a ketone group.